Now we're going to just do a short review of what we already covered in class on bacterial pathogenesis, just to kind of point out the things that you need to focus on when studying. So, one of the first things that we're concerned with is exposure and colonization. And with colonization, so first you're exposed to the organism, and then we have all of our defenses to try to keep it away. But the pathogen itself is going to be trying to get in, and so it can reproduce. Pathogen has to attach. Okay, so that's really going to be the first thing. T. So there are, you should review, a number of different types of adhesins. So there's different ways that the bacteria is going to attach to a cell or a tissue. Right? And you remember just a quick review of some of those. So the bacteria have fimbriae. They can use a particular type of pilus to do that. Bacteria can have specific receptors for certain cell types so that they can attach to them. And some of them are very non-specific too. So just even the glycocalyx. So there are specific and non-specific ways, and these are just a short little summary, there's a lot of others, that bacteria will try to attach to cells that they're attempting to infect and or damage. And so those are some of the main ones that are going to go on there. Once the bacteria has attached, then we have invasion. So invasion of the tissue or a cell, whether it's an individual cell being infected by the bacteria, so the bacteria getting into the cell, or getting between cells, and the cells kind of being damaged in this way. We're going to have release of enzymes. So the enzymes that are going to be released are going to either damage a specific cell or a variety of cells. They can be lytic enzymes causing lysis of cells of a variety of types. Usually they're kind of specific, but there's a phospholipase, for example, that will destroy cell membranes. Hyaluronidase is one of the major enzymes released by bacteria, often called a spreading factor. So it's an enzyme released that will then cause damage to cells and allow the bacteria to spread, right, within tissues. They have collagenase, damaging specific types of tissue, the collagen, and so forth. And there's a whole number of different types of enzymes that can be released by bacteria that are all going to cause different specific types of damage to sometimes specific types of cells or damage in a specific sort of way. Now, one of the main reasons for this invasion or damage is both to set hold within inside the cell or within inside the host organism so that we can get cell division. So the actual infection stage where the cells will multiply. In addition to that, sort of happening like at the same time as invasion is taking place and they're getting prepped for cell division, there's the toxigenicity of the bacterial cell. So we have our endotoxins and our exotoxins. So that endotoxins are attached. For example, one of the primary ones is our LPS, our lipopolysaccharide layer of the gram negative bacteria. Exotoxins are released from the cell. Many of these are proteins. And so you should review the properties of endotoxins versus exotoxins in general. For example, not heat stable, damaged by heat or destroyed. They are heat stable. The endotoxins are not typically damaged by heat. Endotoxins typically cause responses like fever and nausea. The exotoxins typically don't. Exotoxins are usually far more dangerous to the host organism at a very low concentration, whereas the endotoxins usually can be tolerated for a fairly long period of time. And that's related to this concept, this other term called virulence, which essentially means, how much of the bacteria or how much of the toxins released by the bacteria does it take to make ill the host or even kill the host, right? And so sometimes virulence is measured by killing the host using an LD50 value, like how much does it take to cause 50% of the infected organisms to die from the infection. So that's measured by the different types of toxins and their action, whether they're neurotoxins or hemolytic toxins, and a variety of things. And then now, while the bacteria is all trying to do this, they can also be trying to evade the immune response. They could be trying to hide out. They could be trying to resist the immune response. So there's all these other things happening. And meanwhile, you have the immune response coming in with all of your cells trying to identify this organism and tag it so that it can be eaten and destroyed before it gets a chance to divide. So there's the organism itself doing all of these things, and then there are your cells, the immune cells, coming in trying to counteract them at the same time. So just kind of review this process. Make sure you can kind of break it down into attachment, how they attach. So what sort of structures are being used, what sort of chemicals are being used for attachment, the invasion. Enzyme release, what are some examples of some of the main enzymes that might be released to get into the cells and the tissues. Infection, the multiplication, we've already kind of covered that quite a bit. And then the next thing is the toxigenicity. Make sure you can list the characteristics and identify the characteristics of the different types of endo and exotoxins.